Hello, and welcome to the first session in Ovarian Cancer Canada's monthly speaker series. My name is Stephanie Goslin, and I'm the Regional Director for Ovarian Cancer Canada for Saskatchewan and Manitoba. And I'm excited to be your host for the session this evening, Demystifying Clinical Trials. I know that this topic has generated a lot of interest across the country, and I'm very excited now to introduce you to the excellent speakers we have set up for you this evening. Dr. Stephanie Lheureux is the site lead for gynecologic oncology and clinician investigator at Princess Margaret Cancer Centre. Dr. Lheureux holds an assistant professor position in the Faculty of Medicine at the University of Toronto. She is the principal investigator of several international studies and is involved in the development of early phase clinical trials with an emphasis on drug development and translational research in gynecological malignancies. Next, I'd like to introduce you to Valerie Bowering. Valerie has worked at the Princess Margaret Cancer Centre as a clinical research nurse for 12 years and is currently the research manager with the Drug Development Phase 2 program. And last, but certainly not least, I would like to introduce you to Teal's sister, Kathleen Robb, who is living with ovarian cancer and will be sharing her experience participating in a clinical trial. While not tending to her own emotional and physical well-being, Kathy has devoted the past 5 years to supporting other ovarian cancer survivors and advocating for improvements in treatments. For the past 3 years, Kathy has been the top contributor to the Kingston Walk of Hope, raising donations through her own personal social network, and also from the proceeds of the sale of organic produce grown at the farm in southeastern Ontario, where she and her husband have retired. She is a volunteer national moderator for OV Dialogue, Ovarian Cancer Canada's online discussion board for Canadians diagnosed with ovarian cancer, and she also hosts its weekly live online chat, Teal Thursdays. So I will thank you all in advance for taking time to speak with us tonight, and I'm going to pass it over to Dr. Lheureux to begin the session.
So thank you, everyone, for being there tonight with us. It's really our pleasure to be with you and to discuss this specific topic of demystifying clinical trials. So let's talk about clinical trials. So as we just mentioned, we will have a question at the end. So please do not hesitate already to put any question in the Q&A as well. So as the presentation, we will go about different objectives. So the first one will be actually to try to see and define what actually is clinical trial and why clinical trial may be important. Also, the other topic that is very important we wanted to discuss is how we run clinical trial and what that involves, running a clinical trial, and also discuss about the process of clinical trial and that what involved for the patient as well. And we want to give also some example, and we will complete the presentation tonight with some example about what is a clinical trial and what that impact the treatment for patient as well. So we try to answer your question, but please put any question and we will have time to discuss. So first I will start off actually what is a clinical trial. So clinical trial, it's not necessarily dedicated for oncology. A clinical trial is actually possible in different settings. And recently we have discussed quite a lot a clinical trial with a vaccine. With the COVID-19 situation, the access of the COVID-19 vaccine was actually possible because of research and the development of clinical trial. And it's why now we can have access to the COVID-19 vaccine. So clinical trial is not dedicated to cancer specifically. So it's a research study that actually involve people and really try to answer a specific question. So it's really important to see with clinical trial, what is the question that we want to answer? And that may involve treatment. So we can involve drug, for example, drug IV or by mouth or subcutaneous, different type of treatment. Can also involve some intervention. So it can involve intervention by a different procedure or also observation. So there's different type of clinical trial and also can involve different group. So it's a kind of a very large group of people that can be eventually involved in clinical trial. And when we look at specifically cancer clinical trial, it can involve a combination of medication, one medication, a combination of medication. It can also look at radiation because cancer also has some radiation aspect of treatment. So the trial can involve radiation or radiation combined to medication, but also can include some surgery and some specific technique of surgery, or eventually sometimes some specific imaging as well. So that is actually what is a clinical trial, is assessing a new question by new tool, and that involves people. So why actually doing clinical trial in the context of cancer, and specifically here tonight where we are here, and our goal is really to try to see how we can improve ovarian cancer. So why clinical trial? Is any new treatment that we're doing in ovarian cancer is actually based on result from clinical trial. So all the medication that currently is possible in ovarian cancer is actually the result of clinical trial. And how we give the treatment and how we discuss this type of treatment is always based on the result from clinical trial. And clinical trial will be at different time of the disease trajectory. So it can be at the beginning, it can be in the middle, it can be at different time. And it's thanks to clinical trial that actually we have now this new treatment strategy with a PARP inhibitor treatment, with bevacizumab, or all this maintenance treatment that now we discuss. So actually all the current treatment will have been part of clinical trial previously. So I think the next step is to try to see, okay, how we think about a clinical trial and how a clinical trial is born. What is how we actually do a clinical trial. So the first thing that is important to discuss is what is the design of the clinical trial, because when we do a clinical trial in cancer, we really need to have a defined question. So what is the question? And based on the question, we will have different type of clinical trial. And clinical trial, there is different steps of clinical trial. So the stage, as sometimes we talk about stage in cancer, we also talk about stage in clinical trial. And the type of trial will be depending on the drug, depending on the situation. So we'll go further on that. And depending on the question, depending on the stage of the trial, we will also have some specific patient selection. So not everyone can be in specific trial because there's very specific criteria for clinical trial. And another important thing of when you design a clinical trial, we cannot have an undefined number of patients. So we have a specific sample size for patient because we want to have a specific question. So we will need a specific number of patients to answer this question. So it's why before the trial starts, we know that we will need to have X number of patients to really answer the question. And it's not ethical to involve more patients because we don't need more patients to ask the question. So it's why trial will open, close all the time. It's because as soon as we have the number of patients we need to answer the question, the trial will stop to be analyzed. So it's why clinical trial, it's evolving things. For example, on day 1, I will say we have this trial, and on day 10, I will say, no, this trial is no longer available, it's something different. So that changed quite often. And what is also very important is actually all clinical trials are reviewed by an ethics committee. So we cannot do a trial without a review of different people. And each clinical trial will be reviewed by an ethical committee to ensure that this trial, this question, this number of samples is ethical and it's feasible in practice and in clinic. And to ensure that the people who do clinical trial is doing appropriately, these other people that will come to the hospital to actually monitor the trial and to verify everything, to make sure that we're doing everything as part of the trial as it was validated by the ethical committee. So when we do a clinical trial, we will have a lot of people that actually will watch what we do to ensure that we run the trial ethically and appropriately. And another very important thing is as part of the clinical trial, it's all objective and it's the patient safety is first. And the other important thing is the objectivity. Everything needs to be measured because we want to answer the question. So we need to make sure that everybody will have the CT scan at the same time, will have this blood sample at the same time. And that is really to ensure that there is no bias. Because when we do specific study with no specific control or specific rule, there's a lot of bias or other things that can actually impact the result. So the trial is very objective to really ask a specific question. So when we talk about clinical trial, as I say, it's stage of clinical trial, and we call that trial phases. Because depending on the question, we will have different phases of the clinical trial. So for example, in cancer, when we think about trying to improve the treatment in ovarian cancer, we may need to look at the lab of why ovarian cancer progress and why suddenly the cancer cell of ovarian cancer get resistant to the platinum. So then they will have some work in the lab preclinically to try to understand why the cancer cell begin to resist to the platinum chemo. And they may find a specific reason, and they will try to develop a drug targeting this mechanism of resistance. That is a work done in the lab. It usually takes a little bit of time. And then when they develop the drug, then they will assess this specific drug. But this drug is very new. It comes from the lab, it's very new. And even if you test that on the mice, if you test that on different animals, you never know how that will behave in human. So the first step when you have a new drug is to test it in clinical trial, which we call a phase 1. So the phase 1 is really assess a new drug that we don't know much about in human. So this first step of the phase 1 is to see what is the toxicity of this drug and what is eventually the effect of this drug. So we go very slowly to monitor very carefully. So it's why at the phase 1, as you can see here, it's just a small number of patients, and we go very progressively because we monitor very closely. So we can also have a phase 1 when we actually know 2 drugs. We already know 2 drugs, but it's the first time that we will combine them together. So if the first time that we combine the 2 drugs together, it will be also a phase 1 to ensure that there is no specific interaction that we don't know, specific toxicities that will suddenly appear because we combine 2 drugs. So phase 1 is really trying to assess the safety of a new drug or a new treatment or a new procedure. When after the phase 1, we have established the dose, the safety, then we will move to a phase 2. So phase 2, it's a little bit more patient. And a phase 2 is to really try to assess the activity of this drug. The phase 1 show that it's active, a little bit like there's some safety, and have a signal of potential activity. So then they will move to a phase 2 to really assess the activity of this drug in a specific situation. And when after the phase 2, this drug has showed some interesting activity and some potential benefit, this drug will move to what we call a phase 3. And a phase 3 involves much more patient because it's where we really need to be objective in terms of, does this new drug do better than the current standard of care? Because to eventually have an approval of the new drug, we need to ensure this new drug do better than the standard of care, or it's less toxic. We need to really show that this new drug have a benefit compared to the standard of care that we have. So it's why there is a very different step of clinical trial, and it will be very specified which phase the drug or the trial is. Valerie, do you want to say anything about the phase of the trial?
So I think you've covered it quite well, and I think we're going to give an example of seeing some of the drugs kind of go through this. So you'll be able to see sort of the trajectory of this. But I think what's really important on this slide is that clinical trials go from the lab to into a patient, and it takes a long period of time with a lot of people involved throughout the different phases of the trials.
So as you can see in terms of clinical trial and different phases, It's why you will see that the procedure and the different tests that will be required of a patient will be different according to the phases. So for example, for phase 1, we will ask quite often different tests, different blood tests. The patient will need to be seen quite frequently in the hospital to really make sure that we monitor carefully the patient. For phase 3, we have much more data. We have much more information about the side effect, about the activity. So the intensity of follow-up will be less than a phase 1. And when you know the different phase of the clinical trial and what is the objective of the different phase of the clinical trial is where we go in the specific, what we call randomization. So when you have an advanced clinical trial, so usually a phase 3 trial, where we have a new drug, where we know it's the toxicity profile, when we know there's a signal of activity, we really need now to show that this drug is better than the standard of care in a specific indication, because the goal of this type of trial is to have this new drug eventually, if the trial is positive, to change the practice and actually to have that now as a standard of care. But we need to show that there's an improvement of this strategy compared to the standard of care in a specific indication. So as all the trial, you will have specific inclusion and exclusion criteria, and you will have a group of patients that will be in this situation to assess this new drug. And then some patients will have the treatment group, which we call the treatment with a new drug, but we will have a group of patients that will have the control group, and the control group being the standard of care treatment. And it's a random assignment, and neither me, neither Val, neither the pharmacist, neither the patient, neither the family, nobody, we know which treatment the patient will have. And it's where the placebo come in. It's really to make sure that nobody knows, to make sure we can be objective, because there is always bias and we all have our own bias. It's why it's important to have a placebo to make sure that we can be objective, and to make sure that we are objective to be able to compare the new treatment to the standard of care. The patient will be followed the same way. So they will have the CT scan the same time, they will have the blood test the same time to really be objective. So this randomization, when we assign a specific treatment, it's what we call the randomization. And it's really to avoid bias. And the control group, even if you have a placebo in the control group, actually, this is a standard of care. So you have a placebo potentially in the control group, but actually this is usually the standard of care, because as we discussed at the beginning, all trial is reviewed by an ethical committee. So we cannot do something that is not ethical, and we usually cannot do something that is below the standard of care. It's not ethical. If a patient go on a clinical trial, it's not below the standard of care usually. So it's why I think we have a placebo, is to really be objective, but the placebo is usually looking at the standard of care. So that was really regarding the question of the placebo is usually reflecting the standard of care treatment. And it's often when you are in advanced stage of the trial. When you are in a phase 1 or phase 2, usually you on phase 1 for sure, you don't have a placebo, because the phase 1, as we discussed, is really to assess a new drug, the safety of this new drug. So we cannot have a placebo, because the goal, the question of this phase 1 is what is the safety of this new drug. So in phase 1, you don't have a placebo. You can have a placebo in a more advanced trial, because the goal is really to try to see if this new drug do better than the standard of care treatment. So that is usually the concept of clinical trial. And then after, there's a lot of work behind the scene for clinical trial to really actually have a clinical trial. And I think Val will be really the best one to talk about what is behind the scene of how to run a clinical trial.
Thank you. So this slide is just kind of, just go back one. This slide is just a bit of an overview, just to kind of give you an idea of the different levels of authorities that we have to go through when we're running clinical trials. So globally, we have to make sure that everybody that's running the clinical trial, everybody that's participating on the clinical trial as far as staff, are participating as far as good clinical practice. So it's what we call GCP, making sure that we're following all of the rules and regulations. If it's an international study, we're going to have the FDA involved to make sure that it's okay for us to run the clinical trials here. If it's being run here in Canada, we also have to have Health Canada. So Health Canada, FDA, the European Medical Association, so they can come in at any point in time and audit the clinical trials and make sure that we're following the rules and regulations, make sure that we're doing everything that's right, make sure we're doing what we've said we were going to do when we're running these clinical trials. And this is basics for patient safety, to make sure that we are following the protocol, make sure that we are doing what the protocol has set out to do, what the trial has set out to do. The next level that we have on top of that is Cancer Care Ontario. So at a provincial level, we need to make sure that we have all of the drugs that are being given in these trials go through this approval process to make sure that it's okay for us to give these drugs here in whatever center you're in. If you're in, out west, they'll have to go through a provincial level out there as well. And then there's the local. So this is at the institutional level, so at the hospital level. The Research Ethics Board, so this is where Dr. Lheureux was speaking about. This is to make sure that the ethics make sure that we're doing everything properly again. It also needs to go through all of the different departments. So if this trial is going to have patients going for biopsies, we need to make sure that the biopsy area knows what the trial is and make sure that they are on board with it. And then overall, the physician that's running the trial is accountable to make sure that everything at the site level is being done according to the protocols. Next slide. So when you come into to the clinic, wherever you are, whether you're at Princess Margaret or whether you're at your local hospital, there's a lot of people behind the scenes, and you know that in clinical care to begin with. But this slide just kind of depicts where things are at from the research perspective. So Dr. Lheureux already spoke about the preclinical. So it starts with scientists with an idea, with researchers with an idea, then discussing and creating protocols, making sure that this is something that we want to look for, what we want to move forward with. Sometimes the trials will have sponsors. So this includes drug companies. And then you've got your regulatory approvals, which you just spoke about, Health Canada, FDA, the Ethics Board. Some of the sites will also have nurses that run their clinical trials. Some of the sites won't have nurses, but they'll have clinical coordinators that run the trials. Then in the background, you'll also have clinical data management coordinators who take all of the information that the patient-facing people are collecting. So when you come to the clinic and we start asking you different questions, then they take all of that information and they put it into the different databases and making sure that everything is being recorded. And then if it's a treatment trial, then you may have, where you go to a systemic therapy treatment. So you may go to a different department. And then also, if you're running into any side effects or anything like that, you've got other people that would be potentially helping you with those as well. So there's a lot of people that are behind the scenes in clinical trials, and we just wanted to kind of give you a bit of an overview because we do believe that it takes a village to run clinical trials. Next slide. So as we talked about, so you've got your physician, so you may see your physician, you may see your trials nurse or your clinical data coordinator, but you don't necessarily see the pharmacists and the technicians that are behind the scenes making sure that, that medication is right for you, that dosing is right for you. You've got the pathologists that are looking at the tissue, medical imaging, so, CT scans or ultrasounds, all of those types of things, they all have to be involved as well. If there's an intervention, so if there's a biopsy that needs to be done, that whole group needs to be there as well. Radiation, Dr. Lheureux already mentioned this. ECGs, the research coordinators that are doing the data collection. The statistician, so this is one that Dr. Lheureux had alluded to earlier, whereas making sure that the numbers of patients that we are looking for, we have the appropriate numbers. And then, of course, all of the different specialties. So you have the people that are giving your treatments as far as the systemic treatment people, all of the nurses that are in the different clinics. So it does take a village in order to be able to have clinical trials running within an institution. Next slide. So this may be a review for some people. This may be new. I'm not really sure where people are at with knowing about clinical trials, but we just wanted to kind of give a basic overview when you start to hear things like it's not according to protocol, those types of things. So this is just really a basic overview of how clinical trials are done. So protocol is a legal document that explains what will be done in the study, when it's going to be done, and why it's going to be done that way. So protocols can be, anywhere from a couple of pages long to I've seen them over 300 pages long. And so all of that information is something that the trials team, all of the different varying people have to be aware of and make sure that they're following. The protocol gives us a background about the treatment. So if there's been other trials, so if it's already been done in the phase 1 setting, it'll give us some of that information within the protocol and help us with the previous results. Identify side effects. It will also give us some idea of what to do about those side effects. And if you're on a clinical trial, or if you've ever been on a clinical trial and we approach you and say that we need to reconsent you, this is a situation where the protocol has been looked at, new information has come out, and it's gone through all of the extensive reviews for the ethics board and everything. And so then we have to give new consent forms and reconsent patients and make a decision about whether you want to continue to participate in that trial or not. Next slide. So who can participate in clinical trials? So as Dr. Lheureux had said, not every clinical trial is for everyone. So each clinical trial has its own list of criteria that determines who's eligible to participate. And these eligibility criteria can be, very short, and they're looking for just very small amounts of things outlining that, to things that are 20 or 30 items long where you have an inclusion-exclusion criteria. So these types of things can be the type of cancer, your overall general health, previous treatment, medications that you're taking. All of those types of things will play into the inclusion-exclusion criteria. And so that's what we call the eligibility criteria, and that will be in the protocol. You won't necessarily see that, but when you have your coordinator, your nurse in front of you, they're going through all of that, as well as your physician, to make sure that you meet that eligibility criteria. One thing could make you eligible or not make you eligible. It all depends on the history and what it is that specific protocol is looking for. Next slide. So why is the eligibility criteria so strict? So people will say that to me, Val, why can't I participate? I see this great trial, why can't I do it? So 2 important reasons. It's to protect your patient. So it's to protect you as a patient from receiving a treatment that's not safe. It's also to help research identify those people who will most benefit from that treatment. But the basic thing is about safety. We need to make sure that what we're doing and what we're giving you is about your safety. So those are the main reasons why we have all of those eligibility criteria and why they're so strict with them.
Yeah, so in terms of the eligibility criteria, as we discussed, when you are in a phase 1, for example, when we look at the safety, it's very important to make sure that we don't put the patient in the higher risk. So it's why there is very specific exclusion criteria. If you have, for example, immune disease, you will not be able to have a trial with immune therapy. And that is really to try to make sure that we don't increase potential side effects. So it's why there's all these criteria. And in addition now, because we have sometimes a specific question regarding the trial, we need to make sure that we select the patient appropriately to answer the question the best we can. And for example, if we look at ovarian cancer is a big box, and there's different type of ovarian cancer. So when we look at ovarian cancer, the most common type of ovarian cancer is the high-grade serous ovarian cancer. And when we look at the high-grade serous ovarian cancer, biologically, is very different than all the other type of ovarian cancer. So for example, if we do a trial where we put some patient with high-grade serous ovarian cancer, mucinous ovarian cancer, or clear cell, if we mixed all these tumor type of ovarian cancer, we may not be able to answer the question. And we may actually say, oh, it's negative, but it's maybe negative because we enroll more patients with this type of cancer. And maybe this drug was efficient for this type of cancer. So it's why we have now specific criteria. And it's why also we begin to be a little bit more specific now with the type of tumor or specific criteria to make sure that we can answer the best we can the question. And try to identify the best strategy for developing a specific treatment, and to make sure that we don't put patients at risk if we don't think there's a benefit for this type of cancer as well.
So some patients will come in and they'll say, I'm being considered for a clinical trial. What are some of the things that I need to be considering? So I just put together a brief list of some things to think about if you are ever approached to participate in a clinical trial. So you want to discuss with your healthcare team alternative standard treatment. So what are your other options? This is voluntary, it's not mandatory, so we want to make sure that you have all of your treatment options so that you can make an informed decision. Clinical trials are not given in association with other treatments. So if you've already started a treatment, we're not going to take you off of that treatment to put you onto the clinical trial or give it in conjunction with. There are some trials where you will take an oral medication plus an IV medication, but if you've already started a medication, we won't necessarily take you off of it to put you onto a clinical trial because we want to know what the effects of are that treatment that you're currently on. Like all treatment options, clinical trials have possible benefits and risks as far as side effects. So those are things that you need to keep in mind and look through those. And sometimes consent forms can be a little bit scary and daunting when you look at all of the different risks. I say to patients, not every patient is going to get every single one of those side effects on that list. Not every patient will get some patients will get some patients will get others, not everything listed there you will get, but you need to take into consideration and review that closely when you're looking at a consent form. Trial drugs are covered, so patients don't have to pay to participate on the medication, but they will also not be paid. In oncology trials, we do not pay patients to participate in the clinical trials. You will have to cover the cost of other medications. So if you have a side effect, you get nausea, hopefully you have insurance and you're able to get the medications that we're giving you to help combat that side effect. But the trial drug would be covered. You won't have to pay for that, but other medications to help with the side effects won't be. The location. So sometimes we'll have patients that want to come in from different areas across the country, or our patients want to go elsewhere. It just depends on the cost of travel and what that's going to look like. How much you're going to have to travel, are you going to have to stay overnight? Are you going to have to relocate? All of those types of things you really need to keep in mind, specifically because where is your support system? If you're going through side effects or you're having a hard time, is your support system closer to home? Are you going to have access to that if you were to relocate? So these are some of the things that we want you to consider while you're looking at clinical trials. Next slide. The other thing is that when you go onto a clinical trial, you may be given a list of contraindicated medications. So when your nurse comes in and speaks to you and asks you what medications you're currently on, there's a reason for that because we don't want them to interact with the study medications. So if you have that list of medications that you're not able to take, then we ask you not to take them, obviously, for safety reasons, but also to let us know about any medications that you are starting, because again, we need to compare it and make sure that it's safe to take with the study medication that you're taking. The frequency of visits, and I alluded to that as well. Some of the trials, they have daily visits. Some of the trials have once every month, once every 6 weeks. So you need to make sure that you take that into consideration as far as your impact on your daily activities as well. The protocol, so we talked about that. So we are going to be asking you to follow the protocol. Outlined in your consent form is when the study visits are going to be taking place, but we may bring you back more frequently than what's listed on that consent form if we have concerns or if there's something that you have a question about. So although it may be laid out there, you also need to be prepared that you may have to come in more frequently. Potential side effects, we've talked about that. And all data is going to be recorded. So everything that you tell us as far as side effects or medications, all of that is going to be taken into account so that we can report it. And you also need to report that to us. So if you happen to have a side effect and you're worried that we're going to say, hold your medication or decrease your dose, it really is important that you do report that to your healthcare team, to your trials team, because that is a safety concern. We need to make sure that we are treating your specific issue, but also making sure that other patients are aware of this as well. And, if you happen to be admitted to a hospital, then you need to let us know about that as well, because part of that regulatory system that we were talking about before requires us to report any hospitalizations within 24 hours. So we need to make sure that you let us know about that. So there's a lot of constant communication. If you've ever been on a clinical trial, you'll know that you're in contact with your trials team quite often. And then you're also, it is voluntary, as I said before, so you're never required to stay enrolled in a clinical trial. You can withdraw your consent at any point in time. Obviously, you want to have that conversation with your team to make sure, we understand what's going on, but it is not mandatory to stay on trials. Dr. Lheureux, did you want to add anything?
No, we want to know everything about our patient.
Yes. Yes, definitely. Okay, so the screening process. So if you are starting on a clinical trial, just wanted to kind of walk you through what this process looks like. And sometimes, people will say, well, why can't I start tomorrow? I've got this treatment. You've said, yes, I want to start tomorrow. Why can't I start? So just to give you a bit of an overview of what goes on when you do start in a screening process. So essentially you will come in, you will talk to your physician, you'll talk to your trials team, they'll do an assessment, they'll look at that inclusion-exclusion criteria that we talked about before, and then they will discuss the treatment options. We'll talk about clinical trials, we'll talk about the standard of care options, we'll talk about all of that, give you a spectrum of what it is that you need to make a decision. The consent form is given, and I'll talk about that a little bit later. So the consent form is given, you have an opportunity to review that consent form. So some patients will say, Val, I want to sign this today. I don't need to read it, I want to sign it today. So I'm going to say no, because I want you to take it home. I want you to make sure that you have thought about all of those things that we just talked about as far as your consideration of clinical trials. And you're going to take it home, you're going to talk to your family, you're going to talk to whoever it is that you need to speak with, and you're going to take time to make an informed decision. I usually tell my patients, if I've ever been a patient on a clinical trial with me, I usually say, take it home. Don't review it on the same day as your clinic visit. Just think about what it is that we've said, talk about with your family members. And then the next day with a strong cup of coffee or tea or water, whatever it is that you drink, read through it, because it is a long document and you want to make sure that you go through it. And if you have any questions, write those down. Once you've made your decision that you've decided that you want to participate in the clinical trial, you're going to sign the consent form. So you're not going to do that at home. You're going to bring that in and you're going to bring that to your clinical trials team, and they're going to sign the consent form and witness it with you. So depending on what area you're in, that may be a different practice, but just check with them whether they want you to sign at home or whether they want you to bring it back. So that's a question across Canada, wherever you are, what process they follow. And then once you've signed the consent form, there's what we call the screening process. So you start looking at all of that inclusion-exclusion criteria. So we have to do certain screening procedures, and that can take some time. So if, for example, a biopsy is required, we need to book a biopsy, we need to have the biopsy done, and then we need to ship that tissue out to the sponsor. Sometimes we need to wait for that tissue results to come back, sometimes we don't. Sometimes we need to repeat CT scans. So we want, somebody will say, well, I just had a CT scan 6 weeks ago. And I'm going to say, we want to do the CT scan as close to us starting your new treatment as we can, because we want to make sure that we have an objective opinion of where your disease is when we're starting. So we want to make sure that we do all of these things. And that protocol that we talked about will have timeframes as when they want those tests done. So we need to make sure that we follow that within that time frame. Once you've done all of those tests and everybody is happy, per se, that you meet the eligibility criteria, that's when you would be able to start the trial. So it is a bit of a process to make sure that you get through all of the tests and everything that needs to be done. So I talked a little bit about consent, but just wanted to go into it a little bit more. So what is informed consent? So essentially, informed consent is getting all the facts before you make a decision about participating in a clinical trial. So consent forms, again, can be anywhere from 5 pages, and I've seen them up to, I think, about 25 or 30 pages long. So it is a bit of a long document. There's a lot of information in there. You want to make sure you go through that consent form and you're asking questions. So when you come back in, or if you have a phone number to speak to your trials team, call them, ask them the questions to make sure that you have all of your questions answered. Consenting is not just a one-time thing. So I alluded to this before, where we may have to reconsent you. The process continues throughout the trial. You'll be told about any new findings, and we talked about that before. So it's an ongoing process to make sure that you have all of the information you need to continue on to a trial.
Yeah, so it's why it's regarding a question that was asked is it's not because we signed the trial that after it will stay the same, because as we mentioned at the beginning, the trial is monitored very regularly by different people, and the trial sometimes is done in different sites. So all the information that is collected in each site will be then collected in the big center, and that will also be reviewed. And if there is new information that come from the drug or the combination, that will be monitored as well. And if there is new information, it's very important that we get back that to the patient. So it's why sometimes you will have what we call amendment or new consent, because there's new information that is now available. And sometimes as well, with the trial actually can be stopped, because when they review all the data, they say, oh, well, no, we were wrong. Either the drug is too toxic, or actually, no, it's not effective, because the data is reviewed very, very regularly by external people. So it's why sometimes the trial will be stopped before we even thought it will be, because there's new information because of safety. So it's why it's possible that the thing will change if there is any safety concern. Usually if the trial will be stopped early, if there is any safety concern, mainly, because again, it's with the ethical process. We need to make sure that it's ethical. So if we have new information putting the patient at a higher risk, the trial in that case may be stopped earlier than initially planned.
So the consent form provides you with information about the trial and the therapy or the tests that are going through as far as the study is concerned. Confidentiality is discussed, who has access to this information, how's your privacy going to be protected. So as I said, that monitors as far as Health Canada, FDA, or EMA can come in and monitor and audit a study at any point in time. There's also monitors that are part of the study. So they're a separate group of people not associated with the hospital that will come in and again look at all of the data to make sure that everything is being followed according to the protocols. So the patient signature does not mean that you have to stay on a study. A patient can withdraw at any time for any reason. So that's kind of a brief overview of what the consent form looks like, but wanted to kind of just give you a bit of an overview of that there. So after consent, so we talked about this a little bit, there's screening procedures. And so the study is going to have very specific screening procedures. It doesn't mean that if you've been on one trial that you're going to have the same screening procedures and therefore you don't have to worry about it. You don't have to do it again. So some of the screening procedures in this doesn't, it's not all of them. It's not an exclusive list, but we just wanted to give you an idea of some of the screening procedures that need to be done. And remember, these have to be done within a certain timeline. So we've got the CT scan, we've got blood work. And some of the blood work, somebody will say, I just had blood work done last week, Val, why do I have to repeat it? So that blood work may not be the specific blood work that we're looking for this trial. And why are we looking for that specific blood work? Will be because of safety parameters. So we need to make sure that we have that most up-to-date blood work. There's going to be study blood work. So this would be research-specific blood work. Sometimes there's ECG, sometimes there's tissue collection. So this tissue would be removed previously where you've already had a surgery, then we'll look for that, what we call archival tissue. Sometimes biopsies are required, vital signs, physicals. So these are some of the things that you might encounter when you come in for screening procedures. That's also in an interview. So it's not an interview, but it's an assessment with your study team. So your nurse sitting down with you and kind of doing a head to toe basically of where we're starting. So I kind of explain it like it's like a picture. I'm trying to explain where we're starting so that the people that are behind the scenes, all those data coordinators, have that information so that they know what they're putting into the system. So you'll have that interview as well. It's an assessment, and then physicians will also do a physical exam.
So as part of the clinical trial as well, because we develop a specific drug and we're trying to see how best to answer the specific question, more and more now this clinical trial involves tissue request. So either we need to have the tissue that was removed at the time of surgery. But as you can see here, the problem sometimes to use the archival tissue is that the tissue change and the tumor change over time. So it's possible that clinical trial will ask you to do a new biopsy to see actually what new mutation or new change is there at the time of progression, to try to see if we can identify why patient will respond to the treatment or why not. Because even in the schema that we show, these different type of ovarian cancer, even with the high-grade serous ovarian cancer, there's multiple types of high-grade serous ovarian cancer. Some will have this mutation, some will not, and then many other receptors that can be different. So it's why now more and more clinical trials ask a biopsy to really try to see if we can have a better understanding about what is specific to your tumor at this specific time.
So how long will you be on a clinical trial? So that's a question that we get sometimes. People are reading through the consent form and they go, well, how long am I going to get this treatment? Is it, 6 months, a year? Where are we at? So it's going to be specific to your clinical trial. It will say in the consent form. There's going to be specific follow-up. So even once you finish the clinical trial for whatever reason, if you've come off, there will be follow-ups that are done. And so your clinical trials team will reach out to you and chat with you to see how you're doing in the follow-up period. It also depends on the number of patients that are being enrolled. So it will vary from trial to trial. The assessment of toxicity, so this is the side effects. This is going to be outlined in our protocol, and we're talking about, what side effects. So every time you come in, remember when I talked about that head to toe assessment at the beginning? So we're going to do that every time you come in. We're going to add and take away any symptoms that you might be having, symptoms that have gotten better. So we're going to do all of that every time that you come in. We're going to make sure that if you are having side effects, how can we help you manage those? And then some of the trials will have patient questionnaires. These can be electronic, they can be paper. It varies again between trials. The assessment of response. So a lot of times, we go from CT scan to CT scan. So you'll hear your trials nurse say that, or your trials coordinator say that. Basically, the protocol will tell us how often we need to do the CT scan. And the CT scan really still is the best way that we have to know exactly what's happening with the disease on the inside, right? So we want to make sure that we're going from CT scan, depending on when they're laid out in the protocol as far as how often they need to be done. And then, so you'll have a radiologist that looks at your CT scan. Then you'll also have a separate trials radiologist that looks at it. And this is an objective opinion. So it's looking at the specific lesions, and sometimes you'll hear your trials team talking about tumor measurements. So that is specific to the trial. You won't have a copy of that, but people will go through that. Your team will go through it with you just to give you an idea. But it is a very objective to make sure that, we are being objective as opposed to, oh yeah, it looks okay, smaller, bigger, whatever the case is. If there is progression, usually on clinical trials, it means that you're going to stop that treatment. Not always, but it is a conversation that you'll have. But sometimes it does mean that you would have to come off of the trial. The placebo group, sometimes there's an ability to unblind after the patient has come off of the trial. Not always, but sometimes there is. Specifically, if it impacts your future treatment care, then that's that conversation that you'll have with your trials team. So clinical trials in a nutshell, I think you've kind of gotten the point that there's a lot of people in the background. It starts with the question and protocol writing. There's a lot of team members that need to be on board with this. We have to go through the rules and regulations as far as the approval process. Then we're talking to patients, and we're asking patients if they want to participate. Then all of the data needs to be collected. Then there's a statistical analysis that has to happen as far as making sure that everything that we've collected makes sense statistically. And then presentations and publications. So some people will ask for reports out after a clinical trial has been reported, and then all of that data being registered to be able to move forward to the next phase of the clinical trials.
Yeah, and clinical trial, there was also a question. The clinical trial, as I say, it's an answer to a specific question. And the question actually can be raised by different people. And there's a question about who is the sponsor of the trial. You're right. The trial can be conducted in different way of who is controlling the data. And if actually the drug is usually from a company, and actually the company is the one sometimes who run the trial because they really want to assess their drug in a specific indication. So the sponsor sometimes can be the company. And for example, if we do a research biopsy that is required for the protocol, The biopsy is usually sent to the company if the company is a sponsor of the trial, and we may not have any result of this tumor. Sometimes we have, because it's a specific selection criteria for the trial. There's some specific trial that require a specific marker, and that the company will look at it, but will give back and say, you have this marker, you can go on the trial. You don't have this marker, you don't go on the trial. But sometimes we're doing a biopsy and you don't need the result of this biopsy to go on the trial. So in that case, sometimes you don't have the result of this biopsy. It's a research biopsy to try to identify later on why some patient will have response and why not. There is also some trials that actually are academic and run by specific scientists or investigator, so that, for example, some hospital or some investigate can be sponsor of the trial. So it's, for example, we are clinician and we have seen this drug in this specific setting, but we have other patients that we think may benefit in a slightly different indication or combined to a different medication. So we can actually try to be we can be the sponsor of the trial and actually run the trial. Some challenge is to have access to the drug and the budget as well to run a trial, because as you can see, this involves a lot of people. So the budget of running clinical trial can be significant as well. But clinical trial actually is what actually have changed the journey of patients with ovarian cancer. And initially, when we look in, in the 1990s, the main treatment that we have was platinum initially, and then the Taxol. And we were currently quite empiric, and all ovarian cancer have the Taxol and the platinum. And with time and with increasing our knowledge, we begin to say, well, we can't be so empiric and we need to be more precise on how we give treatment. So clinical trial allow actually to be more specific in terms of patient selection. And then in 2010, we have the addition of bevacizumab, because we know that the angiogenesis have also importance in ovarian cancer. So that led to the approval of bevacizumab in ovarian cancer. And more recently was the PARP inhibitor treatment. And that has also led to a change in the practice. Now, what is really our problem is how we overcome this resistance to this new treatment. So now our trial are much more defined in trying to understand the disease biology and the evolution of the disease and the resistance mechanism. So you can see the evolution, but all the new addition was coming from clinical trial. And as you can see, usually the clinical trial is driven by a specific question. And usually the question is because of the clinical challenge. And as you can see here, like initially you have the diagnosis of advanced ovarian cancer, because unfortunately ovarian cancer is often diagnosed at advanced stage. So our goal is usually to do surgery and then chemotherapy, but despite having done this surgery and the chemotherapy, the patient can relapse. And then the treatment was usually to go back on chemo. And because of this pattern, we thought about trying to develop some maintenance treatment. When you do the surgery, you do chemo, we can't see anything, but there's some cell that we don't see that sometimes come back. So how we can try to target this cell that we can't see. So it's why it was all this maintenance therapy that have been developed. But before trying this maintenance with some drug, we had to do some validation to actually show that there's a benefit of doing it. And it's why clinical trials come into place to try to see how we can change the treatment to show there's a benefit. And the benefit is higher than the risk. But it's why we need clinical trial to prove that. And I will just finish this session quickly by giving an example. And it's very biased because it's a positive example. And it's also very important to keep in mind that there's some clinical trials that are negative. And we try new treatment and it doesn't work. So it's why the informed consent is important to know what you commit to, because there's additional tests, additional visit, potential new side effect that we don't know. So it's why it's very important to make sure that your consent form is read and you agree to this process, because by definition, a clinical trial, we don't know. We assess new treatment, but we don't know if it will be positive or negative. So that is very important. I would just take this example of potential impact of clinical trial, and that is a positive trial, but it's not always the case. We have negative trial too. but it's the only way actually to inform the patient, to know it's important to know what works, but also important to know what doesn't work. And just this example of the PARP inhibitor treatment. And again, when you talk about, at the beginning, the lab, and then the phase 1, phase 2, phase 3, and then the approval of the drug is exactly what happened for the PARP inhibitor treatment. And you see the time. We initially discovered the BRCA1 and 2 mutation in 1994. So it took time to say, oh, there's a BRCA1 and BRCA2 gene. Oh, that is involved in ovarian cancer, and how we can target this mutation for treatment. So that takes a little bit of time to develop some specific drug. And then after, we went in different clinical trials. So we have the phase 1, then the phase 2, and then the phase 3. And initially, we don't start at the first line. And we don't start at the first line when it's new drug, because we don't want to compromise the standard of care at the first line. So usually the phase 1 and phase 2 will be at the recurrence time. But when you have a good signal of activity, and when we know really well the safety and some good signal of activity, then you can eventually move to a first line. So it's how the PARP inhibitor treatment was initially from the lab, phase 1, phase 2, phase 3 in the time of recurrence, and then phase 3 at the first line setting. So it took time, and it's just to go through the PARP inhibitor story. So the mechanism is really based on biology. So it's why I really highlight the importance to understand the biology of disease and how it's evolved. So it's why the tissue take more and more importance right now, and to try to really understand the biology. So really the PARP inhibitor treatment target the DNA repair. And if you have a BRCA mutation, you have a defect already in one of the repair DNA. And normally you have another system that can repair. So, the cell can survive. But if you target the other pathway to repair, you can't repair at all, and the cell die. So it's why when you have a BRCA mutation here and you have inhibition of the other pathway, the cell can't survive. If you only give a PARP inhibitor, but you have the other pathways at work, the cell can repair and survive. So this is what is the foundation of the use of PARP inhibitor treatment in the BRCA tumor. And we know also that this pathway of homologous combination is not only BRCA, there's other genes that play a role, but we still need to figure out how to define that in clinic. It's not clear yet. But again, same process. The PARP inhibitor went in phase 1. The phase 1 trial was less than 100 patients in each trial. And the new drug, the PARP inhibitor treatment, was initially assessed as a small dose and slowly increasing the dose to try to really see what is a safe dose that we need to use for after going in a phase 2. So which is a dose that is safe and have some signal of activity that we should use for a phase 2. Then they went to a phase 2. And the phase 2 trial, again, patient selection. We see that actually the patient that have benefit was a patient with a BRCA1 and 2. And then this group of BRCA-like, which we still need to try to better define in practice. We also see in phase 2 that the patient selection was really the patient who have a platinum-sensitive disease, where the best benefits was seen. And it's how they develop the concept of maintenance. When patients have response to the platinum, while trying not the PARP maintenance to maintain this response to treatment. So then this was a phase 2 that was done, comparing the PARP to a placebo. Again, the story of the placebo here. When patients have a recurrence of ovarian cancer, you treat again with platinum-based chemotherapy. The standard of care at that time was to do surveillance after platinum-based chemotherapy at the time of recurrence. You don't give anything. So it's ethical to give a placebo. It's your standard of care. But because this idea of maintenance was beginning to be developed, the trial assessed, if you give a PARP inhibitor maintenance at that time, can you have a benefit? So it's why you have patients who will have a PARP and some patients will have a placebo. And here what they show is actually in this phase 2 trial, having a PARP maintenance was better than having a placebo. The placebo was a surveillance as a standard of care. So yes, this idea of maintenance PARP with this specific patient was important, and specifically the patient who have a BRCA mutation. And how you define the BRCA mutation is on the blood or the tumor. So again, we go back to the tissue and the importance of access to the analysis of the tissue. We also have some idea based on the phase 1 and phase 2 trial about the safety, and it's why it's important to assess all the safety. And Val know the PARP inhibitor really well in terms of safety.
So, I mean, some patients may be on PARP inhibitors that are listening this evening and, watching later. So generally speaking, maintenance therapy, as far as PARP inhibitors, generally well tolerated. That doesn't mean that it's not without side effects. So some people will think that just because it's an oral medication and it's not chemotherapy, that it doesn't have side effects. That's not true. We need to make sure that everybody is aware that there are some side effects with any medication that you take. The most common side effects could be nausea, fatigue, and the hematological toxicities. So this is where we see sometimes a drop in your hemoglobin or your platelet count. You may or may not feel this, but if it's checked with your trials team or with your clinical team, they will be able to see those results and act on them, whether that's with a transfusion or holding the medication, sometimes requiring a dose reduction. It is important that we manage those side effects so that we're able to keep you going on the medications and then also try and prevent them if at all possible. So sometimes for nausea, taking the medication before, so once you start that oral medication, taking something for nausea before you actually get the nausea, sometimes this will help prevent further issues with nausea in the future.
So then after the phase 1, phase 2, phase 3, so the same principle, but we need more data because we need really strong information. So the Solo 2 trial was actually try to have the patient who have a platinum sensitive relapse. Because they have initially identified the patient who have a BRCA1 and 2 were the one who responding the best to the PARP inhibitor, they select initially this trial for the BRCA1 and 2. And patients after the response to platinum-based chemotherapy were randomized, so again, assignment, arbitrary by a computer to a PARP or a placebo, and nobody know who got what. And then after you look at when the cancer recur, which we call the specific process, the PFS, which is the progression-free survival. And again, here you have a benefit of taking the maintenance olaparib was better than to take a placebo for this specific patient population. So then, after they were able to have a look at the overall survival, so also they have shown that you actually delay the recurrence, but also have a gain in terms of overall survival. Then, because they have this data at the time of recurrence, then they move to a first-line setting to say you had the surgery, you had the chemo, and we'll do the same. As a standard of care, when you had the surgery and then the chemo, usually you stop and you are on surveillance. But because of the data we have seen in recurrence, maybe we should actually do this kind of maintenance before and at the first line. It was exactly the question of the trial. You have your surgery, you have your chemo, but you have this BRCA1 and 2 mutation. Patients after response to chemo were randomized between the PARP inhibitor versus a placebo. Same look at the time of recurrence. And again, patient who took, as you can see at the top line, patient who took the olaparib, actually, the control of the cancer was much longer than the patient who actually took the placebo. So definitely, this kind of result of the trial led to the approval of the olaparib at the first line setting for patients who have a BRCA1 and 2 mutation. But what about the patient who actually don't have a BRCA1 and 2 mutation? Because again, it's about which type of cancer. There's another trial that has been done because the question here of this trial was, can the maintenance can be also beneficial beyond the BRCA? So this trial was done, patients who have relapse were treated with a platinum-based chemotherapy. They were responding. So you have the patient who have a BRCA mutation, that was a specific cohort. And you have another cohort of patients that do not have this germline BRCA mutation. Both groups were randomized between having a placebo or the PARP inhibitor maintenance treatment. And as you can see here, the patient who have a BRCA mutation definitely have their disease control much longer than the patient who have a placebo. It was also true for the patient who do not have this BRCA mutation, but have what we call this BRCA-like. So there's some specific test that was done as part of the trial that defined this BRCA-like, which showed that there's a benefit. But actually, even if you are not BRCA-like defined by this test, you still have a benefit. So it's why our research is still to try to understand or better identify this kind of BRCA-like patient, which is a work in progress. But it's why now, at the time of platinum-sensitive recurrence, if you have responding to the platinum at the time of recurrence, you can have access to a PARP inhibitor treatment, which is based on this trial, beyond the BRCA mutation population at the time of recurrence. So as you can see here, to conclude, is that we go from the lab to the clinic. And now in clinic, we have another question, because our patients now have a PARP inhibitor treatment. But some patients, unfortunately, will relapse on the PARP inhibitor treatment. So now our new clinical challenge and our new question is, when I have a patient in my clinic who progress on the PARP inhibitor treatment, what should I do next? What is the next treatment I need to do? So I need to go back to the lab and try to see why patient relapse and what is the mechanism involved in this relapse. So with sometimes what we have done is just an example of a patient who was responding beautifully to the PARP inhibitor treatment, but relapse. What do we find is that actually the tumor changed over time. And she changed because unfortunately cancer are malignant cells and they develop new mechanisms sometimes to resist to the treatment. And for example, this cancer cell have actually be able to reverse the mutation of the BRCA and actually develop another pathway to overcome this resistance. So now we have a lot of work on trying to see how we can target this mechanism of resistance. So it's currently a work in progress, and it's why we need to develop new clinical trial looking at this specific question. And we wanted to conclude about, what about immune therapy? Because we have a lot of questions. Immune therapy has significantly improved the outcome for melanoma, for lung cancer. But again, what about ovarian cancer? It's what we care about. What about immune therapy and ovarian cancer? Unfortunately, there is no immune therapy yet approved for ovarian cancer because the trial that has been done in ovarian cancer didn't show the same as it has been in lung or in melanoma. So right now, in terms of the FDA approval for gynecologic cancer, you can see some immune therapy has been approved, FDA, but even in the FDA, there is no immune therapy approved for ovarian cancer. Some trial has been done, but the trial was negative. Like the way we use immune therapy currently for ovarian cancer didn't show significant benefit. So that is still, again, a work in progress to see how we need to use immune therapy and ovarian cancer. Do we need to combine? Do we need to use different type of treatment? So that is currently a work in progress.
So just to follow up on that, although there is no approved immunotherapy, there are clinical trials that are ongoing that involve immunotherapy in ovarian cancer. And so those will be important results when they come out to see where we can go with the next steps for immunotherapy in ovarian cancer. So access to trials, we get this question quite often. How do you access clinical trials? So a couple of important points that I want to bring up here is clinical trials shouldn't be thought of as a last resort. You know, you've exhausted all of the other treatment options and now you want to look at a clinical trial. There are options that are like that and clinical trials that we do have that could potentially help in those situations. But as Dr. Lheureux had shown before, you know, trying to integrate clinical trials throughout the trajectory of your cancer journey is important because it gives different options throughout the time period. Really important is to think about clinical trials before you're starting a new treatment, if there's time, because we have to remember, you know, if you're not feeling well and you can't wait for that screening period, then, you know, you have to start a new treatment. But if you know, things are moving along fairly slowly and you can think about it before you actually start a new treatment, then that's when you want to think about a clinical trial and speak to your physicians about it. So how do you access it? You speak with your oncologist or your family healthcare practitioner. They're your best people to speak with. Does it have to, there was a question that came up in the chat, does it have to be Princess Margaret? No, it doesn't have to be Princess Margaret. There's great centers all across Canada that run clinical trials. So all of your oncologists will be able to reach out to one of those centers and be able to access what potential options they have. We do have options here at Princess Margaret, but again, there's lots of sites that run clinical trials across Ontario, across Canada. So speak with your healthcare provider about those options and see what would be the best for you. And again, we can't speak to your specific situation, so it's best to speak with somebody that knows your care and knows your history. You can do international searches, so I've put this here, clinicaltrials.gov. This is a website for clinical trials. If you've ever tried to use it, it is a bit cumbersome, so you do have to give yourself a bit of patience. It will pull up every single clinical trial across, if you put in ovarian cancer, you'll get hundreds. If you're thinking about going across the border to the US, you need to keep information as far as that consideration, as far as what we had talked about before, as far as travel, as cost, all of those types of things, to keep that in mind. The other thing to keep in mind is that just because you've looked at it on clinicaltrials.gov, you think that it's open in a site near you, you still need to go through that screening process. So making sure that all of that eligibility criteria is met before you can go onto a clinical trial. Making sure that you have all of your information, I think, is the biggest point. Make sure that you know and have all of your questions answered.
To conclude, and sorry, actually, we thought that we will take less time than the time we took, so I apologize for the time. The best is to come, so stay on. But we just wanted to end by really thanking our patient, because research is not possible without you, and we can making progress thanks to you, because participating in clinical trial, it's a commitment, and actually there's a lot of uncertainty about clinical trials. So first, thank you. And research is key to change treatment and access to innovation. So thanks to you. And it's a scientific and objectivity approach. So any question we can answer at the end, we will stay on. And thank you for your attention.
Thank you both for that excellent presentation, and don't apologize for the time. I know I was certainly soaking in the information, so thank you. I now would like to turn it over to Kathy, who's going to share her experiences being involved in the clinical trial.
So hi, everyone, and Dr. Lheureux and Valerie, thanks for all of the tremendous information that you provided. I think you have really helped demystify the subject for many of us, and it's a very important subject to most of us survivors that are listening to you today. My journey as a clinical trial participant began with a recurrence of high-grade serous ovarian cancer in December 2018, when I recurred following almost 2 years in remission. I'd heard about clinical trials. In fact, I knew a couple of survivors who were either participating or had participated, but that back then, most trials I heard about seemed to be focused on women who were both platinum sensitive, which I was, but also BRCA positive, which I wasn't. So I naturally assumed my only course of treatment for the recurrence would be chemotherapy. Surprisingly, though, when I attended a meeting at the Cancer Center for what I assumed would be some decisions on what and when for chemo, the option of a trial was proposed by my oncology team. It was for the Olaparib Light trial, LIGHT, and don't ask me how they come up with these names, but they're really weird, most of them. A study of the effects of the drug Olaparib, which is a PARP inhibitor, as a treatment in 4 different groups of patients experiencing recurrence and who were all platinum sensitive, but where 2 of the groups did not require BRCA positivity. The group I was being considered for, however, did require an assessment of my original tumor tissue as being HRD positive, meaning there was a genomic instability, but not BRCA. So a sample was ordered from storage and the assessment was done. And within a month, I'd been accepted into the trial and I'd started taking the oral medication twice daily. That was February 2019. For me, the decision to opt for the unknown of the trial versus the tried and true conventional therapy was fairly easy. There was no placebo group to this trial, so there was no fear that I would get a placebo and my cancer would go untreated. And since my recurrence had been caught very early, there was time to assess the effects of the drug and revert back to conventional therapy if needed, without any risk to my long-term well-being. Also, olaparib was not really experimental by then. It had been used for many years in clinical and trial settings, so it was a known medication, and all of the possible side effects had been documented and disclosed to me. None posed any concern since the contract allowed for me to opt out of the trial for any reason. So if the side effects were intolerable and not able to be managed, I could walk away, as I would have had the drug not shown some effect on the cancer itself. Of course, clearly the option of oral medication daily versus chemo, along with the intense surveillance by my oncology team, had huge appeal. And there were few restrictions on me, none of which would impede my enjoyment of everyday life. So giving up things like grapefruit and Seville oranges, I still will never quite understand why that was on the list, was a small price to pay. But the need to increase my attending the cancer center for tests, for blood work and examinations and reports didn't pose a real inconvenience for me. But for others, especially those with children or careers or who live a distance and may need to travel to a cancer center, might need to consider that. And I think the gals mentioned that in their presentation. So from February 2018 to July 2020, when the trial was actually ended by the manufacturer, I responded well to the olaparib. In the next few months, or in the first few months, sorry, the drug reduced my cancer by almost 75%. And after that, maintained it as stable at a size that was almost not measurable anymore. I experienced only one significant side effect, severe joint and muscle pain. That was easily managed with a slight reduction in my dose. Other than that, I found things tasting a bit odd, and some fatigue stayed with me through the course of treatment, but at much less a level of intensity than I experience when I'm on and recovering from chemotherapy. And there was huge comfort and security having the rigor of the required surveillance while a trial participant. I was only required to maintain a log of when I took each dose of the medication and report any issues that they might deem effects of the treatment. But in return, I received monthly checkups, blood work, along with CA-125 assessments, bi-monthly CT scans. Admittedly, given my veins were already tiny and damaged from chemo the first time, I came to rue the days of blood work and the IVs for the scans. But I considered it a necessary evil given the response I was having to the drug. And there were side benefits of being on the trial that we don't talk about. I developed a wonderful relationship with my trial coordinator, a remarkable, funny, and caring gal with whom I still stay in contact. In fact, today I was in chemotherapy and she came by just to sit down and chat with me for about half an hour and keep me occupied, which I thought was absolutely wonderful. And I also discovered a small community of trial patients as we acknowledged each other in clinic reception, holding our little brown bags of empty containers of the medications we were taking that had to be returned each month in exchange for the next month's supply. I never did meet anyone else on my particular trial, but I learned much from the experiences of others, most of which made me realize how lucky we are right now, that there has been so much advancement in research and treatment that, as OCC says, we are beginning to live longer and better lives. After the trial concluded last summer, the manufacturer continued to provide the drug to me at no cost for so long as its effectiveness continued. Sadly, in November, my cancer did begin to progress, and I'm currently now halfway through a new course of chemotherapy to address it. I look back with no regrets whatsoever for opting for the trial. In fact, I'm enormously grateful for having been given the opportunity. Today, I credit it having kept my cancer a low level and me out of chemo for almost 2 years to the very strong response I'm now having from my current chemo treatment. It gave me almost 2 years of a level of well-being that allowed me more enjoyment of that time that I would have had than that time that I would have had under the chemotherapy protocol. I also have renewed hope that research and innovation will surprise me again when I least expect it. and when I need options in treatment down the road. But finally, allowing data collection from my experience will in some small way help to inform changes in treatment, support the release of new drugs, or identify new applications for existing drugs that will benefit the well-being and life expectancy of those who follow in our footsteps. Perhaps in its own way, by participating in the trial, I've been given some purpose as to the why me of this disease. Would I do it again? Well, actually, I have. Clearly, no is not in my vocabulary. I'm now engaged as a participant in BioDiva. It's a study of biomarkers in high-grade serous ovarian carcinoma. With that one, there's nothing in it for me, at least short-term. But the collection of genetic data pulled from the biopsy of my existing cancer tissue and regular blood draws throughout the course of my current chemotherapy will assist in improved understanding of the biology of cancer and understanding the changes in the biology over time that may allow researchers to predict how treatments may work in guiding decisions around the choice of drugs. And if there are any other studies or trials out there where I can bring benefit to the ongoing improvements in treatment for our disease, I guess I have nothing more to say than bring it on. So with that, I'll pass this back to our host to begin the very important Q&A. I see there's some great questions lined up.
Kathy, thank you so much for sharing that. In particular, I loved hearing about the more personal benefits that you have received from the trial, such as the relationships that you've built. So thank you so much for sharing that. And now Kathy is right. We do have some excellent questions. So we are going to get started and reading some of these questions. I'm going to just start with one that I can answer quickly. When will we be available to view this again? As she missed an important slide. So we will have the recording up, we're hoping within a week. So you can check our website and it will be posted on our website. Okay, so I have a question here. If during a clinical trial, you have been placed on the treatment group and you have no disease progression during the period, can you stay on the treatment or must you go off as the trial ends?
That is a good question. So the duration of the trial is often specified in the protocol. And again, it depends on the situation and the context. So for example, if we look at a maintenance in the first line, for example, if we took the SOLO-1 trial or even the PRIMA trial, which was a PARP maintenance in the first line, they have a specific duration. So the maintenance for the SOLO-1 trial was 2 years, and the PRIMA was 3 years. It was initially defined this way. And there's a specific duration in the first line because at that time, we didn't know the long-term effect of a PARP inhibitor treatment. Now we have more data about the long-term, and we have some patients that got PARP inhibitor for more than 10 years. But at that time, when the trial was designed, we didn't have long-term effect potentially of this kind of maintenance. So it's why initially in the first line, they have specific duration. So each trial will have specific duration that is initially set, but that can change over time. So as we mentioned, if actually there's any signal of significant toxicity, the trial may stop earlier than scheduled. But if the treatment was planned for a specific number of cycles, usually after this period, they will stop. But if a patient are on and have, for example, if we are at the time of platinum resistance, and if the treatment was until progression or until significant side effects, normally the patient should be able to continue on until they have benefit. And that is really specified normally in the consent form. So the protocol and the consent form is really the kind of the Bible of what we do for clinical trial.
Thank you, Dr. Lheureux. Next question. I was part of a phase 3 clinical trial until September of this year, until I found out I was part of the placebo group. At that point, the group 1, was canceled. Why would a clinical trial remove the placebo group when the drug is still ongoing?
So, so it's possible that, you know, as I said, the trial is monitored very frequently. And even if as a team, we don't know if a patient have a placebo or the drug, the one who is a sponsor of the trial know it. And it's important that the trial is monitored and look at the data very regularly. And it's already planned at the trial that regularly an external team, a team that is not involved in the trial, will look objectively at the data that has been put in. And if they have any signal of concern, they will actually say to stop the trial. So if, for example, if you know the placebo, which is normally the standard of care, is really not the way to go, they may actually say, oh, we have sufficient data to say that the standard of care is below the new drug, so we just need to stop and then go with this new treatment. So it's the data monitoring that is ongoing that actually guide potential adjustment. Usually the trial go until the number of patients that need to recur, because as I said, the trial is designed as a specific question. So to answer this specific question, we need to be statistically significant. So we need a specific number of patients usually to really answer the question, because sometimes, one patient for any reason will have a good response and we don't know why. So it's why we usually have a sufficient number of patients to really answer the question, to not have any bias for any reason of biology or different reason. So usually there's a specific number of patients. But with all the data, with time, over time that is registered, if there is a strong signal of something, it's possible that this external committee that review will say, no, we stop now. It's possible because of new data coming in.
Thank you. A lot of questions in here about the placebo aspect, which I believe you covered quite well. This is another one here that she says, thank you so much for clarifying the placebo aspect. There is so much misinformation about placebos, and most people believe a placebo is nothing or a sugar pill. I did not know that patients would be receiving at least the standard of care in a phase 3 trial. When fear of placebo stops patients from enrolling in a clinical trial, how do you overcome that perception?
Yes, I can answer, and I think Val will probably have a good insight as well. I think, as we mentioned, trial is really reviewed by an ethical committee. And as a physician, I'm first a physician. So yes, I do clinical trial as an investigator, but my main focus is really caring for patient and ensuring that, I try my best. So yes, I offer clinical trial as an option, but I always discuss the standard of care treatment, because trial is always an option. It's not something mandatory. So when we have a patient, we offer the standard of care or the trial option. And the trial, if there's a placebo, it means that it's ethical to do it. And a placebo is usually when you have a randomization. So you don't have a placebo where it's only one arm. When there's a one arm treatment, there's no placebo. But when there's a randomization, 2 groups of patients, the placebo arm is usually at least the standard of care. You can't put a placebo when you know there's another treatment that will work better than that. And that is really ethical, because our main focus is really to try our best for our patients. So it's why I think there's a lot of work to be done for the placebo, because it's true in a way. The placebo it's not a treatment, it's a sugar pill in a way, but it's not an under treatment.
I think that's really important point, that it's not, you're not missing out by having a placebo. So when we talk to patients and we give the consent form and we're explaining it, sometimes we will get that, I don't want to be on a placebo, I don't want to be part of research, and that's completely the patient's choice. But making sure that patients have the information, making sure that they can have opportunity to go through their questions, and we have an opportunity to answer them, and making sure that they have enough information so that they are comfortable with the decision about participating in a randomized trial that includes a placebo arm. And sometimes what you'll see is you'll have a treatment, so a study medication, with placebo, or a different group of patients that will have, the chemotherapy plus a placebo. So it depends on the trial, and it depends on the design of it and what the medications are. But it really does come down to making sure that everybody has enough information to make the decision that's best for them.
Thank you. We have time for a couple more questions here. This is another one about the placebo. Is it ever possible to advise the participant of their status following the trial? And if the participant was known to be taking a placebo, would that change any future considerations for a similar drug trial?
So as usually we, as I say, when there's a placebo, it's really, we don't know what drug it is. So for example, taking the example of, the solo one, if you have a placebo or the olaparib, we don't know. And if unfortunately the patient recur and have good response to the platinum-based chemotherapy, and if there's a BRCA mutation, I think it's important to know if the patient have, a PARP or not before. So that really impact the treatment care, and that is really the indication to actually go back to the sponsor of the trial and say, I need to know if the patient want the drug or not, because that will impact the future care. And if that impacts the future care and what I will do for my patient, I can ask to unblind, because that will impact, because we need to know. But if the unblind of a placebo will not change what we do, that actually may not be possible to unblind.
Some questions left here. Would someone qualify for a clinical trial without knowing if they are BRCA positive or negative?
So again, it depends on the criteria for the trial. So the trial will have specific criteria. So some will require, some will not. But usually now, most of the trial will require at least the tissue to know. Even if the patient don't know, the trial usually will request some tissue to actually have this information. So it's why most of the trial now require tissue to actually try to have this knowledge about the BRCA status. And now we're trying to go beyond the BRCA status to actually go more in detail about the specific type of cancer. So it's why we ask usually tissue as part of the trial. And initially, when we look at the initial trial with the PARP inhibitor, it was not necessarily focusing on BRCA. It's after we say, oh, the patient actually respond best with the BRCA because they had tissue. So they were able to look back. Because I think the tissue actually can sometimes provide important information about how to guide treatment.
Thank you. Last question here, and I apologize if you've already answered it. What is the extent of the information I can get for how I'm responding to the clinical trial? Can I know about specific results from my biopsy, blood tests, etc.?
So as part of the clinical trial, you will always know what is required for treatment decision. So for example, as a treating physician, I will need to know if you have benefit of the trial or not. I don't want to continue a trial if you don't have benefit. So it's why, as part of a clinical trial, you will have CT scans very regularly. So it's why something also we have is why I need to do so often CT scan. As Val mentioned, our main decision is based on CT scan. We don't base on CA125 because sometimes it's not as specific and it can change for variable reason. So we really base our treatment decision for response, not based on the CT scan. So it's why we do the CT scan often regularly and at the same time for everyone to ensure that there's benefit to continue, because it's an experimental drug. So we don't want to put the patient at risk if there's no benefit. So we do CT scan very regularly to ensure there's benefit. So that you will know the result of the CT scan, but the CT scan will be looked also by another radiologist to use a specific objective assessment of response, which we call RESIST, which is a specific objective way that every radiologist in the world can use to make sure that we can compare results. Because you can look at CT scan sometimes very differently as well, and you can see that how we measure that, it can vary. So these are specific criteria that the trial used to make sure that we are objective. Regarding the biopsy, again, it's possible that if the biopsy don't change the treatment, or if the biopsy don't change your eligibility for a specific trial, you may not have the result. If the trial is sponsored by a company, the tissue is sent to the company, and if that not change your result for the treatment or for eligibility, you may not get back the result.
It's the same thing with some of the blood work as well. So a lot of the trials, specifically the early phase trials, will have a lot of blood work that's required, and you won't necessarily get results of that blood work. It's important when you look at the consent form, because it will lay it out in the consent form, or if there's an optional consent form, that will all be laid out as far as what's available and what results you will get. And you can ask your study team as well. But a lot of times, if it's not going to impact your eligibility specifically for the trial, but they're going to use it for future research or to do a deeper dive into that tissue, then you may not get the results of that. So it may be specifically research-based, and you wouldn't necessarily have those results given back to you.
And the assessment of response as well is also based clinically. So even if for any reason the CT scan don't show any change or any progression of the disease, if the patient don't feel well or actually clinically progress, then we can stop or further trial. And even if the CT scan as part of the trial is booked after 2 months, for example, if the patient don't feel well within this 2 months time, or if we have any concern, we can always do a CT scan before, or actually stop the trial before. We don't have to really say, oh, the CT scan was booked only 2 months after the start of the treatment, I can't do one before. I say, no, we can do what is appropriate for the patient. If the patient, after, for any reason, 1 month of treatment, feel worse, find that the fluid is building up, we will do a CT scan much earlier to actually see what's going on. And we don't necessarily, have to wait this time to assess if there's any signal of clinical symptoms that we need to investigate.
Thank you. That's all the time we have for this evening. And I really, again, just want to thank Val and Dr. Lheureux and Kathy for the excellent information that you've given tonight. I know it has certainly, for myself, helped to demystify clinical trials. We've gotten lots of excellent feedback, so thank you again for participating in our inaugural session of Ovarian Cancer Canada's monthly speaker series, and have a great evening. I also wanted to let everybody else know that we will be running this series every month. We have our next session is already booked, so you can save the date. It will be on Wednesday, March the 24th, again at 7 p.m. Eastern. We will be joined on March 24th by Jennifer DeWitt-Turner, who is a registered psychotherapist and certified care spiritual practitioner. Jennifer's session will focus on dealing with anxiety and uncertainty, as well as emotional healing.